So last chapter, when we went through the menstrual cycle, we went through the steps assuming that the egg or ovum did not get fertilized. Well, in this chapter, chapter 28 on pregnancy and human development, we go through the steps where the ovum does get fertilized and the steps of pregnancy and early human development. So here we're looking at relative size from fertilization all the way up to a 12-week fetus, and you can see a dramatic increase in size. That's about 5 inches, 90 millimeters, versus something almost microscopic. We see that growth, that dramatic growth from those first 12 weeks. So we go from fertilization to a 1-week conceptus as the cells are dividing, and then a 3-week embryo, all the way to an 8-week embryo. So those first 8 weeks after fertilization or post-fertilization is termed the embryonic period. So fertilization to 8 weeks, and then weeks 9 through birth, or around 38 weeks, would be the fetal period. One more point to add here is that if we're talking about pregnancy, we typically we do add 2 weeks to this. So an 8-week embryo would be at the 10th week of pregnancy. Not because the fertilization occurred 10 weeks ago, that was 8 weeks ago, but they measure pregnancy from first day of the last menstrual period because that's measurable, and that individual would know when that day was, where we don't necessarily know when this day was here. So pregnancy is measured from the first day of the last menstrual cycle. So it would be 2 weeks past these weeks that we see here. So a 12-week fetus is actually 14 weeks of pregnancy. A little confusing, but that's how they measure pregnancy versus these are embryonic and fetal periods. So typically we think that fertilization is when the sperm combines with the egg or ovum. That's technically not quite correct, because fertilization, even though the sperm and the egg have united, fertilization does not occur until the chromosomes from the sperm and the chromosomes from the egg combine. So that is a fertilized egg, which we call a zygote, and the zygote is the first cell of the new individual, which will contain 46 chromosomes, 23 from mom, 23 from dad, and there's our first cell, which we call the zygote. And for fertilization to occur, there has to be quite a bit of timing that takes place. So it says intercourse must occur within a certain time period because the oocyte is only viable, only lives for less than a day, about 12 to 24 hours after ovulation. So if you think back, we learned that fertilization typically occurs in the uterine tubes. That's because after ovulation, by the time the egg travels through the uterine tube and into the uterus, well, that would take too long past that 24-hour period, so that oocyte isn't viable at that point, which is why fertilization occurs in the uterine tubes most often. And something to remember as well is that ovulation timing can vary between cycles. So it could be at a different time during the cycle. We learned that those first 14 days in that uterine cycle aren't necessarily 14 days in one female compared to another. Sometimes it's unrelated to hormones. Ovulation can occur, it's possible, without the correct sequence of hormone events. So also, fertilization is most likely in 1 to 2 days after ejaculation. So sperm can survive for up to a week. However, it's their first 2 days after ejaculation that it's most likely to cause fertilization. So it's important to note that most ejaculated sperm don't make the trip to join with the egg. It's about a 12-centimeter trip, but the overwhelming majority of them do not get there because some leak out immediately, some are destroyed by the acidic vaginal environment. Only a small portion actually make it through the cervix, which is the entranceway or the neck of the uterus. Uterine contractions will disperse sperm as well. There's phagocytes that destroy sperm. So out of the millions of sperm that are ejaculated, only a small percentage, a few thousand, sounds like a lot, but it's just a fraction of the million that reach the uterine tubes where the egg is. But the sperm cannot penetrate the oocyte until it's capacitated. What that means is its motility is enhanced and the cell membranes become fragile enough to release hydrolytic enzymes. So kind of to explain this, if you can recall a picture of the sperm, in the head there's that acrosome. We'll see a picture of that in a second. And inside that acrosome are hydrolytic enzymes that are going to allow the sperm to penetrate the barrier to get into the oocyte. So the membrane becomes fragile enough to release these hydrolytic enzymes from the acrosome, and that has to be timed correctly. The secretions of the female tract help weaken and thin out that membrane around the acrosome. So if it thins out too early, it's not going to be viable. If it doesn't thin out when it gets there, it cannot penetrate. So only those sperm that have this timing correctly, where their membrane is broken down enough right when it reaches that the oocyte, will have a chance to penetrate and cause fertilization. And also, sperm have receptors to find the oocyte. They don't swim around blindly. They sense fluid flow direction, temperature. The oocyte releases chemicals, so sperm have these receptors, like olfactory receptors, that help direct them towards the location of the oocyte. So here we see hundreds of sperm cells approaching the oocyte. On the oocyte, we have the corona radiata. Remember the granulosa cells of the corona radiata. And then on the interior of the corona radiata, we see in this gray color here, that's the zona pellucida, which is a lining or a barrier preventing the sperm cells from entering. So the first step, the approach, we see the sperm cells can weave their way through the granulosa cells until they get to this zona pellucida. So here we see a sperm cell approaching the zona pellucida, and it's actually going to bind to receptors. They're being labeled right here, all those little dots. It binds to a receptor on the zona pellucida, and not just one. There's going to be many that do that, even though only one is shown here, binding to the receptor, which causes a series of reactions. But in this step right here, step 2, this is called the acrosomal reaction, because what happens is the enzymes that we mentioned a few minutes ago from the acrosome get released, which will start to digest holes inside the zona pellucida. And again, from this sperm cell here, there's going to be other ones that get in and do the same thing. So the enzymes released from the acrosomes are going to help bore through the zona pellucida. And we can see that happening right through here. So this is step 3, binding. Well, there's also receptors on the oocyte. And the sperm will bind to the receptors on the oocyte, which allows the next step to proceed, which is called fusion. And that's where the plasma membrane of the sperm and the plasma membrane of the oocyte fuse together. And when that happens, well, the sperm will eject its contents inside of the oocyte. Now, this is not fertilization yet, because the genetic material in the nucleus hasn't met the genetic material of the oocyte yet. But before we do that, we need to prevent other sperm from entering the oocyte, because if that happens, that would not be a good thing, because there would be too many chromosomes. And of course, we need the correct number of chromosomes for fertilization to be viable. So that would be called polyspermy. That's when more than one sperm enters the oocyte, which is not good. So to prevent or block polyspermy, the first thing that happens is these receptors that we see here that bound the sperm originally, well, those receptors are shed, and then we get a reaction, which we call a zona reaction, or cortical reaction, or a zona reaction, which there's chemical events that occur, which causes exocytosis, which you see in this blue cloud here of chemicals which harden the zona pellucida. And with a hardened zona pellucida, and you can see here no more sperm can get through, even with their enzymes that they use to break through. So now we have one sperm that has fused with the oocyte, and we can see its genetic contents ejected into the oocyte. Now there's the sperm nucleus, there's the genetic material of the oocyte, the 23 chromosomes, but remember, this was stuck in meiosis 2, in metaphase of meiosis 2. So there's the chromosomes, and there's that polar body from the prior division of meiosis 1. So now that we have the sperm that's penetrated and the sperm nucleus inside the oocyte, well, then the oocyte will go ahead and complete meiosis 2. And we get that, like we said, that second polar body forming. I think I said that was from meiosis 1. No, that happened because we just divided and we have that second polar body after that second meiotic division. Then the sperm and the ovum nuclei will swell. They'll get a little larger, and we call these pronuclei at this point. We can see the centrioles starting to emit their mitotic spindles right there. And what's going to happen, they're going to go through something like mitosis at this point. We can see the spindles now forming between the centrioles. So the DNA in each pronucleus replicate, just like it does before mitosis happens. They make copies of themselves as they would normally do in interphase in a somatic cell. So these pronuclei of the sperm and pronuclei of the oocyte replicate. There's the mitotic spindle, and now we sort of have this nucleus of all of the chromosomes from the sperm and the oocyte mixing together. So now for the first time, the genetic material from the sperm and the oocyte intermix. That is fertilization. So fertilization just occurred right here. They haven't mixed yet up in step 3, but in step 4, the pronuclei come together. You get intermingling of the chromosomes. Now that is officially fertilization. So we can call this cell now not a oocyte, not a sperm cell, but a zygote or a new individual. Now this zygote is ready to divide. The first cleavage division will happen, but that's where we'll begin the next video. We're going to go from a zygote to a blastocyst, but again, we'll continue that story on in the next video.